Before we can talk in any sort of coherent way about extraterrestrial life, we have to understand terrestrial life. And before we start to speculate about how common or uncommon the processes that lead to life are, we have to understand what those processes are. So we have to understand what processes led to the origin of life on Earth. And this is something that unfortunately we do not understand very well at this point in time. What do we know about life on Earth? We know that it's been here for a really long time. Why am I having so much trouble today? Jeez. We're gonna edit that out. Earth has been here for about 4.5 billion years as a solid terrestrial planet with solid surface, liquid oceans, and an atmosphere. Our best fossil evidence indicates that there has been something alive on Earth for almost 3.5 billion years. So the majority of the history of life on Earth has been occupied by life. The direct evidence for living organisms on Earth goes back about 3.5 billion years. There's some slightly sketchier evidence that goes back even older than that, maybe 3.8 billion years. So as soon as the Earth was solid and cool and had an atmosphere and ocean that were stable, there was stuff living in it, which means the origin of life happened relatively quickly on our planet. That being said, the history of life on Earth, which spans nearly 4 billion years, is mostly the history of simple single-celled organisms and bacteria. For the first 3 billion years of the history of life on Earth, until about 1 billion years ago, there was nothing alive on the Earth but bacteria, single-celled microorganisms, and very simple multicellular organisms. Which means the history of life on Earth is mostly the history of very, very simple organisms, not plants and animals and things that we think of. If we extrapolate this to thinking about extraterrestrial life, the thing we might conclude from that is most inhabited, quote-unquote, planets that we're going to find, if we ever find any inhabited planets, but most of them will be inhabited by single-celled organisms and very, very simple forms of life, because that is what life was on Earth for most of its history. Our modern theories of biological evolution tell us a lot about the process of diversification and change in life that happened over the 3 billion year history, from the formation of single-celled organisms to the formation of multicellular organisms to the evolution of plants and animals and the diversification of the whole biological tree of life. What the theory of evolution does not tell us about is the origin of life, how life actually got started on the Earth. Evolution, by natural selection, is a theory of how biological change accumulates when organisms inherit beneficial traits from their predecessors. Evolution doesn't work until there's something alive to inherit something. Evolution doesn't happen until there's some kind of organism that is reproducing. The theory of evolution says nothing at all about how that process gets started. Much like the Big Bang Theory only explains what has happened since the universe started expanding. It does not explain what started that expansion. So what about the origin of life? What do we know about it? The short answer is we really don't know much at all about it. This would be a very short lecture if I stopped there, but I could stop there. As an astronomy professor, I could just say, biologists haven't figured this out yet, so we don't know. We know some things. We have some ideas, so let's talk about what those ideas are so we're not left completely in the dark. Even as long ago as Charles Darwin in the mid to late 1800s, imagined that life started in the water, and it started with chemical reactions. Darwin said we can imagine a warm little pond with ammonia and phosphoric salts and light and heat and electricity that cause chemical reactions to happen that lead to things like proteins and more complex changes. He was very vague about what these changes were and what these chemical elements were. Darwin didn't even know about DNA. It was the 1800s. So Darwin had no chance of explaining how life got started. He didn't even really understand the basic chemical processes that are responsible for heredity. That wouldn't happen for another generation, almost another 100 years. When do we start really thinking seriously about this process? 1924, Haldane and O'Parin coined this phrase, primordial soup. The idea is the early oceans of the Earth would have been a rich soup of organic molecules like amino acids, and these amino acids would gradually combine together into a more and more complex chemistry, and that chemistry would eventually lead to what we would think of as life. Now, how do you test a hypothesis like that? We don't have a bottle of ocean from 4 billion years ago. How can we know what the chemistry, the prebiotic chemistry of life would have been like? One idea that some scientists came up with in the 1960s is to try to recreate it, to create a chemical system that would be similar to the atmosphere of the early Earth. The famous experiment that studied this process was called the Miller Uri Experiment in the 1950s and 1960s. What Miller and Uri did was to, in the lab, try to create the Earth's early environment in miniature. So they took a container, they filled it with certain gases, they applied certain forms of energy to it, and they tried to examine what sort of chemistry would spontaneously arise in such an environment. So they took things like methane and ammonia and hydrogen and water and just put them in a container and then subjected them to things like electricity, ultraviolet light, heat to stir up those chemicals and get some chemical reactions happening. What they found was that when they did this, they would get simple organic molecules, chains of carbon atoms, like amino acids and their precursors, and that this material created a sort of organic goop inside of their vessel that is exactly the sort of thing that was imagined to be the substances of this organic soup, amino acids and long chains of carbon atoms. Here's a picture of a run of the Miller Uri experiment showing some of the organic goop. The organic goop has a sort of orangey brown color that should be familiar to us by now, that we see in the outer solar system on the surfaces of many objects like Europa, Neptune's moon Triton, Pluto's moon Charon, Pluto itself. This organic stuff, this orangey brown coloration that Carl Sagan dubbed tholins, collectively to describe long chains of carbon atoms forming simple organic compounds created by ultraviolet light falling on methane and ammonia and nitrogen on the surfaces of icy bodies in the outer solar system. The chemistry that causes these compounds to arise is actually very, very similar. And what was shown by the Miller Uri experiment is that organic compounds can spontaneously be created if you've got the right chemicals and a source of energy. In the case of the outer solar system, the energy is mostly ultraviolet light. On the early Earth, it could have been heat, lightning, ultraviolet light, all of these things together. That's a long way from life, but it is the building blocks of life. Now, one of the assumptions that went into the Miller Uri experiment was a reducing atmosphere, an atmosphere that had a bunch of hydrogen, molecular hydrogen in the atmosphere. That assumption has been called into question since the 1960s. What do we mean by reducing? Just, this is some chemistry, you don't need to really know this, but when we talk about certain chemical reactions, the chemical that gives up its electrons is the reducer, and the chemical that takes up the electrons is called the oxidizer. So when hydrogen and oxygen combined, for example. Oxygen is the oxidizer and hydrogen is the reducer. The idea that the early atmosphere of the Earth would have been rich in hydrogen is probably not true. And if you take your Miller Uri container and you fill it with a bunch of oxygen, the whole thing kind of fizzles and nothing happens. If you put oxygen in your Miller Uri experiment, the organic goop doesn't appear. Now, the early Earth's atmosphere probably didn't have a lot of oxygen, but it probably didn't have a whole lot of free molecular hydrogen either. So one of the things we have to figure out in order to make an educated guess as to what the chemistry of the early Earth would have been like is to understand what the chemical compounds present on the early Earth would have been like. Were methane and ammonia there? We're not so sure nowadays. There was lots of methane and ammonia in the early Earth's atmosphere. One of the reasons that we want to study other objects in the solar system other than the Earth is to understand the chemistry of these primordial bodies. It turns out if you use carbon dioxide and nitrogen, two things that are present in our atmosphere, and there was probably way more carbon dioxide 3 billion years ago than there is now, and you add minerals like iron and carbonate minerals, which would have been falling on the Earth due to bombardment from meteors and comets, it turns out you do make the organic goop again. So the kind of organic molecules you get are very sensitive to what minerals and what gases and what liquids are present in your little container. The reason we are keen to study objects like Titan, objects like Triton, objects like meteors and comets is to resolve some of the uncertainty about the atmosphere of the early Earth. Studying an object like Titan might tell us something about what the early Earth's atmosphere was like. Studying objects in the Kuiper Belt might tell us something about the composition of the meteorites and comets that were falling on the Earth early in its history. So studying primitive bodies in the solar system, bodies that have not been shaped by life for 3 billion years, might be able to tell us something about the composition of the early Earth, and that will tell us something about how this chemistry might have unfolded. Right now, those are barely more than guesses. Now, what do we know about life on Earth today? We know that the basis of all life on Earth is DNA, this long chain of base pairs that stores the genetic code that allows our cells to make different proteins and do different things inside of our body, and which carries the hereditary information that allows a DNA strand to make copies of itself, which allows cells to make copies of itself, which allows you to make a copy of yourself, and allows every organism on the Earth to reproduce, and that's what life is. So this complicated molecule had to start somewhere. How could that be? The what's sometimes called the Central Dogma of DNA involves DNA, RNA, and proteins. DNA acts as a template to create RNA. RNA acts as a template to create proteins, and proteins are what do all the stuff in your cells that needs doing. And the process of kind of heredity and information storage happens on the DNA side, and the process of kind of metabolism and being alive happens on the protein side. And if you think of DNA as a template for creating proteins, and if you think of proteins as being essentially the chemical metabolism of the cell, what allows it to be alive, a lot of what proteins do is act as enzymes, that catalysts for chemical reactions in your cells. If you think of DNA as being the replicator side of that, and proteins as being the metabolism side of that, each of these things needs the other in order to work. And this creates something of a chicken and egg problem for the origin of life. If DNA is the replicator and the instructions to make proteins. And proteins are the metabolism of the cell, which creates the chemical reactions that allow us to make more DNA molecules. How could either one of these things have happened without the other? The answer is they couldn't have. They both had to happen at the same time, and that is hard to imagine. Until you realize that the thing that sits in the middle of this process is the RNA molecule, ribonucleic acid, links the DNA side of this equation with the metabolism and protein side of the equation. Look, I'm not a biologist. If a biologist watches this video, probably cringe at a lot of what I'm saying being extremely oversimplified. Yes, I'm extremely oversimplifying this because we don't have time to take a whole month's worth of biology and learn about the genetic code and transcription and all that business. I don't know about all that stuff. But the basics go like this. RNA is a chain of nucleic acids, just like DNA. It's a ribose kind of backbone, sugar backbone, that are connected together in a long chain with bases that form sort of a code that allow the RNA molecule to act as a template for making proteins. The base pairs for the bases for RNA are C, A, U, and G. Base pair, similar to the base pairs to DNA, except the U is different for DNA. It's a T. Genetic code aside, you can think of RNA as a sort of set of instructions for piecing together amino acids into a protein. Okay. As such, RNA, can be thought of as an informational template. That the sequence of bases on a RNA molecule can be thought of as a series of instructions for building a protein. So RNA is similar to DNA in that sense. You can think of it as a set of instructions. You can also think of what RNA does, though, as acting as a catalyst. That the presence of RNA causes certain amino acids to join together into proteins. And being a catalyst is what proteins do in a cell, sort of. When they're acting as enzymes, a protein is a catalyst. This gives rise to something called the Rna World Hypothesis, which is that before life properly got started on the Earth, before there was DNA in cells and organisms, there were RNA molecules that were capable of reproducing by catalyzing their own formation. That an RNA molecule arose that could basically catalyze itself, that could cause amino acids to assemble in a certain way that made another RNA molecule. This process of autocatalysis, or an autocatalytic loop, chemical loop, is sort of what life is, right? You are a big chemical reaction that every couple months makes another copy of you out of different molecules, everything you eat and breathe. So you can think of life as a catalytic process, and we can imagine the first precursor to life just being a chemical reaction that catalyzed its own continuing production. So the RNA World Hypothesis suggests that RNA started as the first sort of living molecule, and only later did DNA become a part of that process. And we can imagine an increasing cascade of chemical complexity as RNA began to catalyze more and more complicated things. So, first of all, what do we mean by a catalyst? Imagine a toy universe that only has two kinds of atoms, atom A and atom B. We can imagine assembling A and B into different molecules, an AA, an AB, a BA, and a BB. And those chemical reactions happen depending on how much an A atom wants to stick to another A atom, or an A atom wants to stick to a B atom. In chemistry, a catalyst is a chemical that causes another chemical reaction to happen more quickly. It doesn't participate in the reaction, it just kind of pushes that chemical reaction along. If we imagine this toy chemical universe with just two kinds of atoms and all these different molecules that you can make out of these two different kinds of atoms, and then add into that chemistry the possibility that these chemicals can catalyze the production of other chemicals. So suppose the chemical AAB helped to catalyze the production of AAA out of AA and AA, right? So the more AAB you have in your soup, the more AA will combine with AA to make AAA. Okay, so it's a catalyst for that reaction. It helps to push that reaction along. It lowers the energy of that reaction and helps it to happen. Now imagine that AAA catalyzed the production of some other reaction. Say the addition of B with BA to make, B with BB to make BBB. Suppose that was a catalyst that happened in this system. And suppose the presence of BBB catalyzed B and AB to combine to BAB. In other words, if we imagine different molecules acting as a catalyst for different reactions, we can imagine a situation where the presence of a certain molecule catalyzed the presence of another molecule, which catalyzed the production of another molecule, which catalyzed the production of another, which catalyzes the production of that first molecule again. So AABBBB catalyzes the production of AAB, which catalyzes the production of AB, which catalyzes the production of this, which catalyzes the production of this, which catalyzes the production of that. So it has catalyzed its own production through a loop, a complicated loop of chemical reactions. We call this complicated loop an autocatalytic loop, and it kind of looks like a metabolism. It kind of looks like a DNA molecule using a bunch of cellular machinery to make another DNA molecule in a very, very, very oversimplified, highly simplified form. So this kind of autocatalytic loop, some scientists think could have been a precursor to what we think of as life. It is a self-sustaining chemical reaction that catalyzes its own continuing production. The presence of AABBBB will make more AAB, which makes more AAA, which makes more triple B, which makes more BAB, which makes more AABBBB again. And one of the candidates that some scientists think could be the first autocatalytic chemical is RNA. That what RNA was on the early Earth was the first chemical to catalyze its own production. We can write a computer simulation with a made-up fake chemistry, with strings of letters and numbers taking the place of real atoms and molecules. And we can make an autocatalytic loop. That doesn't mean that this really happened on the Earth 4 billion years ago. What it does mean is that it's mathematically possible for a certain chemical to catalyze its own production, at least in the abstract, at least with fake made-up computer chemistry. We can think then, once this happens, of the catalytic chemical as being the replicator. It is making more copies of itself the way a DNA molecule does. And we can think of the catalytic loop as being the metabolism that is the living process of the cell. Is this life? Well, that's a really good question. Is a chemical that just makes more copies of itself alive? I don't know the answer to that. Now, there are some scientists who think that with a complicated enough system, autocatalysis is mathematically inevitable. That if you have a complicated enough mathematical system, you will eventually, complicated enough chemical system, you will inevitably wind up with some sort of loop by which one chemical catalyzes its own continued production. once that chemistry gets complicated enough. But nobody has ever succeeded in making this happen from scratch. Nobody's done a Miller Uri type experiment where we just throw a bunch of chemicals together at random and then have an autocatalytic loop emerge from that. But we are getting close to that. Recently, experiments have been done to create a strand of RNA that is able to make copies of itself from a soup of organic material and amino acids, just by catalyzing its own production. This didn't happen by accident. This happened by scientists creating a specific chain of bases to build an RNA molecule that could do this. But we've only been working on this for 50 years. The oceans of the early Earth had hundreds of millions of years to develop complicated chemistries. So we don't know that this is what happened on the early Earth. What we do know is that chemical processes like this are possible. It is possible for RNA molecules to spontaneously catalyze the production of more RNA molecules. So the Rna World Hypothesis is an idea for the kinds of chemical things that could have been going on the early Earth. There's also research showing that RNA can spontaneously form long chains in the presence of certain minerals. For example, on the surface of clays, RNA, or the bases of RNA and the ribose sugars, will spontaneously form these long chains of RNA molecules. So we can actually see how what we normally think of as just rocks could have played an important role in pushing along the chemistry of the early Earth by being the place where these RNA molecules grew into these long chains until eventually they were able to catalyze the production of more RNA molecules. But again, we don't have a step-by-step picture of how we went from soup of amino acids to self-replicating RNA molecules, and we don't even have a clear picture that that is in fact what happened. What we have is a bunch of disconnected facts about prebiotic chemistry. We know that amino acids can spontaneously form under a lot of physical and chemical situations, many of which probably similar to what happened on the early Earth. We know that it's possible for something as simple as an RNA molecule to catalyze its own production and thus make copies of itself similar to the way that DNA eventually would inside of living organisms on the Earth. We know that certain molecules like lipids, just fats, will spontaneously form bubbles in water because one end of the fat molecule pushes away water molecules and one end attracts them, and so the fat molecules tend to be pushed into a sphere when they're immersed in water. And these lipid bubbles are not too terribly different from cell membranes. And we can imagine a lipid bubble enclosing a bunch of amino acids and RNA molecules that would be something like the very first cells. Right now, all these ideas are disconnected pieces that don't form a coherent picture of the step-by-step process that led to life on Earth. We may never know exactly how life on Earth got started. What astronomy can help with this is by giving us examples of other places in our solar system and elsewhere where these simple pre-biological processes might be happening. Places like Europa, places like Ganymede, places like Enceladus, places like Titan may have chemical processes that are similar to the prebiotic chemical processes that were going on Earth. So when we talk about conditions on places like Europa, Ganymede, Enceladus, Titan that are maybe conducive to habitability, does that mean there's life there? No. Are we confident that there's life there? Most scientists aren't. But even if all we find, say we go to Europa, say we drill down a kilometer beneath the ice on Europa and send some probes down there to sample the oceans, even if we don't find anything alive, if we find complicated organic chemistry, that by itself might teach us an awful lot about life on Earth. I've said many times in this course by now that we cannot make a generalization from one data point. And right now, the only data point we have about the origin of life is Earth. And that data point is so far away in Earth's past that we don't actually know that much about it. So even the tiniest discovery of organic molecules in the tail of a comet, organic molecules on the surface of Pluto, organic chemistry in the atmosphere of Titan, every little thing we learn about organic chemistry in our solar system is a possible signpost towards a process that could have been going on the early Earth. So the more we understand these pre-biological processes in our solar system and beyond, the more we understand life on Earth. Now, the one thing that would give us infinitely more information about life would be to actually discover life somewhere else on some other planet, whether it was similar to life on Earth or very, very different from life on Earth. Either way, we would learn a huge amount about what sorts of life is possible. For now, we're constrained to imagining only life that is like life on Earth because we don't really have any more examples. So as we go on and talk about astrobiology, we'll try to distinguish between imagining life as we know it and possibly imagining life as we do not know it on objects that may be very, very different from the Earth.